All right, I want to welcome everyone to today's episode of the Polyploid webinar. Today we have two great speakers who will talk to us about different aspects of polyploid biology. Up first is David Gerard, who is currently an assistant professor at American University. David obtained his bachelor's in mathematics and molecular genetics, followed by a master's in statistics at The Ohio State University, before he pursued a PhD in statistics at the University of Washington. At the University of Washington, he studied multivariate analyses of tensor-formatted data sets under Peter Hoff. Following his PhD, he worked with Matthew Stevens at the University of Chicago as a postdoc, where he studied RNA-seq differential expression analyses in the presence of unobserved batch effects, which all sounds like very scary things I probably should worry about on my own. Recently, though, he's developed an interest in polyploid genetics and is well known for one of his packages that I think many of us who work with polyploid data, this package updog, which we're going to hear more about today. And so today he's going to tell us about the updog, our package for genotyping polyploids. With that, I'll let David take over and go from there.
All right. Thanks, Mike, for hosting this webinar series. Thanks for everybody for tuning in. I'm going to be talking about some things that I did with a bunch of folks. So I'd like to thank Felipe Ferrao at the University of Florida, Augusto Garcia at Sao Paulo, and Matthew Stevens at the University of Chicago. Particular thanks to Felipe for helping me out. I have a statistics background, and so I really rely on biologists to guide my intuition and make sure that I don't make a fool of myself. So particular thanks to him. Today I'm going to be talking about this software or some ideas that we had for genotyping polyploids. So we took a Bayesian approach, and whenever you do Bayesian research, there are three things that you need to consider: the likelihood, the prior, and the way that you're going to come up with the posterior distribution. So I'll talk about those three things if I have time. But let's start out with the problem. We're looking at biallelic SNPs. Say one SNP, the reference allele is denoted A, capital A, and the alternative allele is denoted lowercase a. And the goal is to determine the number of A alleles that each individual has when we're given a bunch of read counts as data. So we really had in mind reduced representation sequencing technologies when we were developing this method, like GBS or RADseq. And this is always the cartoon I have in my head when I'm talking about these sorts of assays. So suppose that this is a diploid individual, and we're interested in the SNP where there's one C and one T. And the data how we get the data in order to determine that this individual has one C and one T, which is the goal, is that we chop up the genome into small reads, and then we make many, many copies of these small reads. We sequence these small reads, and then we map them to some reference genome, some known typical example of the genome from some species. And then we look at this location, and we count up the number of reads that this individual has. So this individual had one T and two Cs. And so based off of these two Cs and this one T, the goal is to determine that this individual has one C and one T. But of course, we're interested in polyploids, but it's the same process. We use the same process. We count up the number of reads for each allele, and then the goal is to determine that this individual has three Cs and three Ts. So I'm going to present this genotyping problem in a graphical way. This scatter plot represents a single SNP, and each point is an individual. On the x-axis, we have the number of reads that an individual has that contain the alternative allele, lowercase a. And on the y-axis, we have the number of reads that contain the reference allele A. And then these lines that jut out from the origin represent the different genotypes, the different dosages that are possible. So this is a hexaploid individual. I believe it's sweet potato. And then the idea for genotyping is that each individual belongs to one of these lines. Each point belongs to one of these lines, and then they vary about these lines according to some noise. And so genotyping is making the determination of which points belong to which line. So sometimes making this determination is really easy. Like for this point up here, it's very close to this line. This line corresponds to a dosage of 5. And so it would be pretty easy to make that determination. For other points down here, it would be a little bit more difficult. And this is a relatively well-behaved SNP. And so we'll talk about other SNPs where we see issues and how we accommodate, how we deal with those issues. So first, I'd like to present a common likelihood that people have typically used in the past, and I'll introduce some notation. So let xi be the read count for the reference allele. So the number of reads that contain cap A, ni is the total read count for individual I, and so ni minus xi is the read count for the alternative allele, lowercase a. Let pi be the proportion of an individual's genome that contains cap A, so it's the dosage divided by the ploidy. Here k is the ploidy. So for a tetraploid, for example, if you have no cap A's, your dosage is 0 and pi is 0. If you have 1 cap A, your dosage is 1 and pi would be 1/4. So a common likelihood that people have typically used in the past is they say that the reference read count conditional on the total read count and the proportion of an individual's genome that contains cap A is binomially distributed with size ni and success probability pi. So if this were just the whole likelihood, doing genotyping would be pretty easy. You could estimate the PIs by maximum likelihood, say, just iterate over the possible values of PI and choose the one with the highest likelihood for each individual. But there are a bunch of issues with using this likelihood. We'll talk about 3. The first issue is that these sequencing technologies, they can produce sequencing errors. So a read is mistakenly called a cap A when it should have been a lowercase a, or it's mistakenly called a lowercase a when it should be a cap A. So this is a well-known problem. Lots of people have come up with the solution for this. 2 less well-known problems with these data is that sometimes one allele might be more likely to be observed than the other allele. We call this ascertainment bias. And this might be caused through the amplification step or through the read mapping step. And so we'll talk about how do you deal with allele bias. And then overdispersion is a problem that we've seen in these data. So overdispersion is greater variability than what you would expect under an assumed model. And so the binomial distribution we found is not dispersed enough for the sequencing data. And so how do we account for overdispersion in these data? So this is, so for the rest of this talk, all the data comes from the study by Shirasawa and company from 2017. They used a hexaploid sweet potatoes. It was RADseq data. These individuals were from an S1 population, 1 generation of selfing. They're all siblings, and so that's the reason why you'll see only some of the genotypes show up and not all of the genotypes. It's because the possible genotypes are constrained based off of the dosage of the parent. So I'll show you a bunch of plots of these genotype plots and to demonstrate issues that these data have, and then I'll show you the way that we accounted for these issues. So first, this is a relatively well-behaved SNP. It appears that there are 3 clusters of individuals, so a cluster of 4s, a cluster of 5s, and a cluster of 6s. So, I fit these data using the updog software under the assumption of 0 sequencing error, so no sequencing error. I didn't allow there to be any error whatsoever in the read calls. So most of the genotyping looks pretty good. These are the estimates that updog gave, except for over here. It seems that there are a few black dots when there should be blue dots. So some individuals were called to have a dosage of 5 instead of 6. So this can occur if you don't assume that you have, when you assume that you have zero sequencing error, because if you have mostly cap A's in your data, but one or two lowercase a's, that automatically excludes any other dosages. It automatically excludes a dosage of 6 if you have one or two lowercase a's. And so you mistakenly identify these individuals to be heterozygous instead of homozygous. So lots of people have noticed this. The way that you can account for sequencing error is to add a parameter that directly models the sequencing error. So to modify the success probability from pi to be the f of pi and epsilon, where f of pi and epsilon is the probability of A and no error plus the probability of lowercase a and sequencing error. This shows up in the likelihood, and so we can estimate it using the read counts, and it's been used by lots of other folks. So I'd like to present what the sequencing error does visually to these genotyping plots. So as you increase the sequencing error, what happens is that these lines, which are the expected counts, kind of squish to get closer and closer to each other. You don't really see sequencing errors this large. I just did it just to demonstrate how that changes these lines more obviously. So these are some empirical estimates of the sequencing error rate from a bunch of SNPs from that Shirasawa study. And most of the time, the sequencing error is pretty small, but it's still important to account for it so that you don't mistakenly identify some individuals as heterozygous instead of homozygous. Less well known as an issue for these sorts of data is allele bias, which I'll demonstrate with this genotyping plot. So at first glance, it looks like there are two clusters of individuals. So a cluster of individuals with genotype 5 and a cluster of individuals with genotype 6, right, at first glance. But these individuals are all offspring. They're all siblings from a generation of selfing. And so if a parent had a dosage of 6, you would expect to see all the children to have a dosage of 6. And if the parent had a dosage of 5, then from Mendelian segregation, you would expect to see offspring in proportions of 1 to 2 to 1 for a dosage of 6 to 5 to 4. So we'd expect to see actually a large cluster of 4s among here as well. So what really seems to be happening is that there are 3 clusters here, a cluster of 4s, a cluster of 5s, and a cluster of 6s, and then they kind of shifted towards the y-axis here. They're not falling on the lines that they should be falling on. The reason that we see this in the data is due to allele bias. So some alleles might be more likely to be observed than others. So let's talk about how we can model this allele bias. So let's say that F is the probability of an A after you adjust for sequencing error. And then let's add some notation to say what the probability of observing a read given each allele is. So let's say that the probability that we observe a read, after the mapping step, after everything, given that it's an A is C, and the probability that we observe a read, given that it's a lowercase a, is D. So what we're able to model is not the probability of A, but the probability of A given that we observe it. We have to condition that we've actually observed the read because that's the data that we have. But we don't have that. We have the probability of A and we have the probability of observing a read given A. But we can come up with the probability of A given that we observe the read by just using Bayes' rule. And so the probability of A given that we observe a read is equal to the probability of observing a read given A times the probability of A over the probability of observing a read. And so when we substitute in terms, we get that this is equal to CF over CF plus D times 1 minus F. And then we can divide the numerator and denominator by C to get F over F plus D over C times 1 minus F. And we can replace D over C with a new parameter H to show that the probability of an A given that we observe a read really only depends on this ratio. It really only depends on the ratio C to D. So if you, in order to account for allele bias, what we did is that we just replaced the success probability with BISSE. So BISSE is this complicated function, f over f plus h times 1 minus f, where f is the probability of an A after adjusting for sequencing error rate. So we only introduced one new parameter. This parameter is the ratio of these probabilities of observing a read given the different alleles that are possible, where this ratio is equal to 1 when there's no allele bias, and it's less than 1 if there's a bias for the reference allele, and it's greater than 1 if there's a bias for the alternative allele. So this shows up in the likelihood. And so we should be able just to estimate it from the read counts. So in the same way that I demonstrated how the sequencing error changes these lines, when we change the allele bias, we shift these lines towards either the y-axis if there's a bias towards the reference allele, or towards the x-axis when there's a bias for the alternative allele. So these are some empirical estimates from that Shirasawa data set. They're centered at 1, which means no bias, and there tends to be more bias towards the reference allele than towards the alternative allele. But there are a large number of individuals that have a bias very far out towards 0.5 or 0.6. And so you could show up in a situation where this is the real situation, but you assume that the situation is here, and this can bias your results. You can get genotyping errors if you don't account for allele bias. The third issue that we saw in these data is overdispersion, so more variability than you would expect under a binomial model. So this is just another SNP from the Shirasawa data set. And I drew out these curves to be 95% intervals. So we would expect 95% of all observations to fall within these curves. So this is the 2.5 percentile binomial distribution. This is the 97.5 percentile of the binomial distribution. And then the orange lines are the dots are the individuals that fall outside those curves. And in this SNP, it's about 20% of the dots fall outside the curves. And you see this all the time in a lot of these SNPs. And in other SNPs, it's way worse. Like, I don't know if this is, there's any information in the SNP, but it's clearly overdispersed beyond what a binomial distribution would suggest. So one way that you can model overdispersed counts from a binomial is to use a beta binomial distribution in the same way that you use a negative binomial distribution to model overdispersion in Poisson counts. We introduced a new parameter, the overdispersion parameter tau, and we model the counts as beta binomially distributed with mean success probability BISSE and this new overdispersion parameter tau. When tau is 0, then we get back to the binomial distribution, and when tau is greater than 0, then we allow for more dispersion in the binomial. And so this shows up in the likelihood, and so we can estimate it using the read counts. So these are these curves again, these 95% curves, but for varying levels of tau. These are some empirical estimates for the overdispersion parameter from that Shirasawa data set. And there's a large proportion of SNPs that have overdispersions greater than 0.01, which is this situation right here. But if you're using the binomial model, then you're really assuming that you're in this situation. And so this can really lead you to being confident in your genotyping estimates than what you should be if you were properly accounting for overdispersion. We took a Bayesian procedure in order to estimate all of these parameters that we introduced. We have a likelihood. Let's say that, let's put all of our likelihood specific parameters that we just introduced into this vector eta. So the sequencing error rate, the overdispersion parameter, and the allele bias. And then let's say that the prior distribution is indexed by some vector theta. And so the goal is to come up with a posterior distribution over genotypes. Let's map out what this means. So what we have is a likelihood. So f of x given y and eta that we've just derived the beta binomial distribution conditional on the sequencing error rate, the allele bias, and the overdispersion parameter. So this maps a probability of the sequencing reads given the individual genotypes. But what we're really interested in is the probability of the individual genotypes given the sequencing reads. So if we have the probability of the individual genotypes, then that means that we can come up with genotyping estimates, either by coming up with the ones that are the most probable, or by coming up with posterior mean genotype estimates. But in order to do this, using Bayesian inference, you need to have some sort of prior knowledge, some prior distribution over the individual genotypes. And we'll say that that's f of y given theta. So theta and eta are unknown. We have to know them in order to be able to do this inference scheme. But these two, so the probability, but we know the probability of the individual genotypes given theta, and we know the probability of the sequencing reads given the genotypes. And so this induces a marginal likelihood of the sequencing reads given the likelihood specific parameters and the prior specific parameters. And so we can estimate them by maximum likelihood. So let's estimate them by maximum likelihood. They get little tildes. And so now that we know these, we can calculate this posterior distribution. The scheme is automatic at this point. So visually, what this looks like is that we start at some initial conditions. So the likelihood specific parameters are represented on this plot according to the location of these lines. And then I'm letting in this one slide alone the width of these lines to represent the probability of each dosage. So here, this orange bar, the thickness of this orange bar indicates that the probability of 5 is very high at these initial conditions. And then in this scatter plot right here, we have the log likelihood, the marginal log likelihood, this term right here. At each iteration as we iterate through the optimization scheme. So as we move through the optimization scheme, we increase the log likelihood until we get something that looks reasonable. So the this Bayesian inference scheme requires you to choose some class of prior distributions to optimize over. And if you are doing Bayesian analysis, the first two things that you naturally gravitate towards are things that are either uninformative or things that are very flexible. So in this case, that would be the discrete uniform distribution. So just say that each genotype has an equal probability of occurring, or the general categorical distribution, which says that each genotype has some probability and they're unconstrained except that they have to sum to 1. So this doesn't work too well in practice. So this is a genotype plot. It seems that this SNP is fairly well behaved. There are just 3 clusters by I, cluster of 4, 5s, and 6s. But if you use the discrete uniform distribution, you get something that looks very unreasonable with a lot of bias and a lot of unintuitive genotyping genotyping results. So, and if a similar thing occurs if you use the general categorical distribution. Yeah, great, 2 minutes. All right. So the reason why this occurs is that a principled empirical Bayes approach would first integrate over the uncertainty in the likelihood specific parameters before estimating the prior distribution, but we don't do this for computational reasons. And so that makes things a little bit more fragile, that's particularly when you use the things that are either too constrained, like the discrete uniform, or things that are too flexible, like the general categorical. And so there are suggestions for how do you come up with priors of classes of intermediate levels of flexibility. What we ultimately decided on was something that was to choose a prior class that was proportional to a normal distribution, and it worked really well. Since I'm running out of time, I'll just go through an example of what the software looks like. We went through all of this theory, but in order to actually use the software, you don't need to know that theory. All it takes are two matrices of read counts. So a matrix of total counts, total read counts, and a matrix of read counts for the reference allele. You insert it into one function, the multi-dog function. You tell it what the reference count matrix is and size and the total count matrix is. You tell it the ploidy of the species. So we assume that all individuals are the same ploidy. You tell it the prior class to use. The proportional normal distribution is the default. And the software allows for parallelization. And so you can tell it the number of cores to use to increase the speed of the genotyping process. So there's a plot method that's available. You just write plot, and then the object that you saved the output to and the SNP number that you want to plot, and it gives you these genotype plots that I've been using throughout the presentation. The output comes in the form of two data frames. So a data frame of SNP-specific information and a data frame of individual specific information. So the SNP-specific information contains things like the estimated allele bias, the estimated sequencing error rate, the estimated overdispersion, other things like an estimate for the proportion of individuals that you genotyped incorrectly. This is a posterior summary that is very natural to describe the quality of a SNP. So we can estimate the proportion of individuals that we misclassified. And so you might want to throw away SNPs that have too high of a value of the proportion of individuals misgenotyped. And then the individual specific SNP contains your genotype estimates. So it contains the posterior mode genotype, the posterior mean genotype, and then the probability of each genotype. We're not the only genotyping software out there. There's a lot of them. They all have advantages and disadvantages. Our big advantage is that we're the only ones that estimate adaptively the allele bias and the overdispersion for each SNP. Polyrad has some really cool ideas on how do you handle population structure in LD. EBG has some really cool ideas on how you account for inbreeding. And then there are a few really good non-sequencing genotypers. I really like Supermassa and FitPoly. FitPoly works surprisingly well, even on sequencing data. It's just that when you get larger levels of overdispersion, it becomes less stable. But it's a pretty good genotyping software as well. So this is where the software is. It's just up on CRAN. You can install it from R in the usual way. This is my email address, and these are the two papers where we developed the ideas here. All right, so thank you so much.
All right, well, thank you, David, for a wonderful talk. We have time for a couple of questions. If folks have them, send them in the chat box, and then we'll have you fire up your video like last time and ask a question. We also will have time afterwards, after the next talk, for more questions. All right, well, we're going to let people ruminate on that, I think. I have a couple of questions, but I think we'll go ahead and let Rosana get started.
Okay, that sounds good.
Switch over. So thank you, David. And yeah, if you could just stop screen sharing, there we go. And then Rosana can take over. All right. For a very early morning way out in the Pacific, Rosana is coming to us as an assistant professor at the University of Hawaiʻi. And I appreciate folks here as we stretch here across the globe from the folks watching in Pacific all the way to Europe. I think we got a 12-hour time difference there. Really looking forward to this talk as well as we continue our day here in statisticians turning to polyploidy. Rosana began her work she was as a master's student in statistics and probability at the Center of Research in Mathematics in Mexico. And after that, she switched to evolutionary biology where she worked on studying the mode and tempo of polyploidy and angiosperm diversification at the University of Florida. Following her PhD at Florida, she was most recently a postdoc with Emma Goldberg at the University of Minnesota, where she worked on models of diversification. Rosana's work focuses on the development of phylogenetic comparative methods to understand trait evolution and diversification across the angiosperms. And today she's going to tell us about some really interesting work that combines all of this stuff to study the interaction among polyploidy or ploidy breeding systems and lineage diversification. And with that, I'll let Rosana take over.
Thank you, Mike. And before starting, I want to tell a brief story. So Mike was the first associate editor that handled my first author paper. So he gave me my first break on the polyploidy world. So I'm very thankful for that. And I feel like he's giving me another break now during pandemic. So thank you for organizing the webinar. So I mean, we are all here because we all care about polyploidy. So you shouldn't be surprised, right, that when I tell you that polyploidy in plants creates major opportunities for evolution and ecology. One of our most beloved examples in the Hawaiian Islands is the evolution and the adaptive radiation of the silver swords, which are hypothesized to have a whole genome duplication before the long dispersal to the islands, and definitely adapted so well to the different microhabitats that we have in the different islands of the Hawaiian region. Anyways, so one of the big questions in this field is whether or not polyploidy is good or bad in the long run. And when we say good or bad in the long run, what we really want to know in macroevolution is where it occurs in the phylogeny, and then how many lineages happen after it occurs, right? So for example, if polyploidy were to happen here, we will say, well, there's a lot of species that have a stem from that whole genome duplication or polyploidy event in general, and maybe because there were many prongs to this, like masking deleterious alleles or very good response to environmental changes, breeding system changes. And all of this have very well discussed in the literature with many field and lab experiments. But if polyploidies often happen in lineages that are going extinct, then we can think about the cons, which also have been really discussed in our literature in our field, right? With like minority cytotype exclusion or time-consuming mitosis and meiosis, right? Finding a multivalent chromosome is not an easy thing to do. So how do we average these pros and cons in large phylogenies? And the one way that we like to set up this question is in macroevolution is whether or not polyploidy is a result of more speciation and less extinction than diploidy. Interestingly, right, I'm not the first one to ask this question. There's been a long and especially very recent history about how to address this question. And it turns out that the models and the botanist assumptions do not match when we're thinking about question. At the beginning, the modelers found that polyploidy is not that great. It turns out that diploids have faster speciation and less extinction than polyploids, and this comes from two very influential papers, which Mike is also an author in, from first author Itai Meiros in 2011 in Science, and then later in New Phytologist in 2014 with the same set of authors. But of course, this initiated a really nice discussion in the literature with the botanists wondering, why are you finding this? Like, what is happening? We know that in highly diverse plates, there's a strong polyploidy ancestry, and why is it not reflected in these results? And really nice contributions from Doug and Pam Soltis and New Phytologist also in 2011. And then most recently, Jacob Landis, he found mixed results, but he found an association with the frequency of polyploidy events and the diversification rate. But of course, my perspective is slightly different because I come from a statistics background, and I am thinking about, these two words and the interdisciplinary work that I need to do. And I always tend to think that the intuition of botanists and people that are looking at these plants is really well rooted. And at the same time, I see the evidence in the models, and there's no question that in the models, this is coming up over and over. So during this debate and this discussion, in these 9 years, a lot of the statistics and the way we collect data has radically changed. We have better and larger phylogenetic trees thanks to the really nice work of systematists. We have larger aggregation of data sets. We see this with the tree of sex, for example. We see this with major studies on woodland herbaceous and climate, GBIF approaches, global change approaches. But we also have seen really big leap, I would say quantum leap theoretical advances in the modeling. And if you think about it, the first model to address these questions in my field started in 2006, which is one of the models that I'm gonna discuss. And the most recent one comes last 2 years, 2019, 2018. So in 14 years or so, we have radical change in how we approach this. So whatever was said back in 2011, it can be reevaluated because of these changes. So my default was to look for a clade where we had a lot of information, and that clade was the Solanaceae family for me. It has really nice and beautiful genera, a lot of them of economical importance, and it was data rich. It has a really nice time calibrated phylogeny by Tina Sarkinen. It also had 14,000 records of chromosome numbers that come from many different sources, but mainly from CCDV, the chromosome count database. And then before this project, I spent a long time figuring out how to curate those records and doing work with an undergraduate student who allowed me to visualize a lot of the chromosome number distributions. And if you want to see them, you just have to go to icrom.com. It's the work of Rodrigo Rivero in 2019 too. So matching the phylogeny and these records, we ended up having 651 taxa on the tips of the Solanaceae tree that had ploidy and breeding system information. At the beginning, I didn't start with breeding system, but this is gonna become more evident as I progress. So in this work, what we try to focus is in 29 models, which seems like a lot, and this slide is overwhelming, that are how the traits affect diversification in different ways. So instead of trying to focus on this slide that is really ugly and really tiny in your screens probably, what I want you to think about is the logic behind these models. So one can focus on the trait itself and ask whether it's polyploidy and ploidy as value, breeding system, or both affecting diversification. But also there's a layer in which we only can focus on the states, so diploid, polyploid, self-incompatible, compatible, or we can focus on noise in this large phylogeny that is not associated to the states I observed, or it might be a mixture of both noise and the states that I observed. So as I'm telling you the story of these models, and if you're inclined to read the paper, a good strategy is to print that slide with the models and think about this strategy on going trait by trait or combining them versus what combination of states and noise creates diversification in Solanaceae. The first model that we tested is a very famous model that is called the binary state speciation and extinction model. This is the model that was proposed back in 2006. And in polyploidy world, that means that we have two states, diploid in blue and polyploid in pink. And we have a rate for speciation of diploids, a rate for extinction for diploids, a rate of polyploidy, how we evolve from diploid to polyploid, then a rate for speciation for polyploids, and a rate for extinction for diploids. So we fit this model that is birth and death, for those of you who are inclined to statistics, in the phylogeny with the trait data of ploidy. And what we found was exactly the same that Itai Mayros found in 2011, that diploids have faster net diversification than polyploids. And when I said net, I mean the speciation minus extinction rate. And it's very significant, right? So I recovered the exact same result that they obtained by using a different data set, more data rich, and with a very well investigated phylogeny. So the piece of my heart that is botanist was a little heartbroken. I was wondering whether it was something that I did wrong or something from the data, why I'm not getting this intuition that the botanists have in a clade where it's packed full of polyploids and diploids. So there could be two reasons. One is that BISSE model is not appropriate to approach this, or maybe my sample is not good enough. But since I'm a modeler, I was going to attack number one. And it's possible that the sample still is not good enough because there's about 3,000 species in Solanaceae and I only have 600, right? So as a modeler, I decided to approach number one. Also in the literature, there has been a lot of discussion about this BISSE approach. And the main discussion that has been occurring over these years is what is happening with the null hypothesis for BISSE model. It assumes that speciation and extinction are the same. But for anyone here that is building phylogenies and is a good systematist, we know that the rate at which lineages accumulate in our trees vary so much. At some points are faster and some points are really, really slow. And just considering a null hypothesis where the rate is constant on creating these lineages over and over is really silly, right? So two very influential papers from Davis in 2013 and Rabowski and Goldberg in 2015 really show strongly that there's high type 1 error in the BISSE models. However, if I might say, well, it is not only high type 1 error, rejecting often this null hypothesis in favor of having different rates. What is really happening, the real problem is that we have a null hypothesis misspecification. Or null should be that diversification accumulates differently in the tree of life. And then we should compare that null versus the model that actually associates that diversification rate to our states and see if it compares to be better. So a couple of people that are really also very intelligent figure out this in 2016. So you can see that before 2016, we couldn't really figure out if BISSE was wrong. And the model they propose is the hidden states speciation and extinction model, or HESE, that a lot of you probably have read in different articles. And in polyploidy world, this is pretty similar to BISSE, as you can see here, but you see a subindex A, which specifies that there's a new hidden state, 8, that could be associated with the diploid and polyploid states, and also a value B or a state B that can be associated, and they could be back and forth between those hidden states. And by doing so, what we add is this noise of an unobserved state to the diversification process and try to parse out what is true signal versus noise or versus just random change in the diversification in the tree, in the lineage diversification in the tree. So when we fit that model to that same data, what we observed is that blue and pink posterior distributions here are completely overlapping, and it is just dark and light, dark on the left and light on the right, that are not overlapping, which pretty much emphasizes that it is not the states diploid or polyploid by the noise on the diversification process, which is making the difference. So BISSE was not an appropriate model, and it is not that diploids are diversifying faster. Both polyploids and diploids are diversifying at the same pace, and it is something else. So when I saw that, my botanist heart again broke, but I started wondering, like, is the hidden state a real trait that I could measure, or a biological process? And is it worth searching for? But I was at the right time, on the right place, working with Emma Goldberg. And Emma and Boris Igich have this very beautiful paper from 2010 showing how self-incompatible lineage diversify faster than self-compatible. So that's when I started collecting the data on breeding systems from their original data set and adding some new observations, and replicated their analysis, finding here in green with BISSE that self-incompatibles have faster net diversification than the purple self-compatible net diversification. And again, this paper from 2010 can suffer the problem of null hypothesis misspecification. So it was necessary to do here, again, a hidden states, which was the next step. So the next step was precisely fitting this hidden states model. And what we found here is that there's a significant difference with self-incompatible versus self-compatible lineages in both hidden states A and B. And this was a strong evidence that, yeah, there's noise and variation on lineages, but the breeding system itself is very important in the diversification process. So as we think about this, what I've done so far is fitting these hidden models to their ploidy and breeding system. However, a plant is both polyploid and self-compatible, or diploid and self-incompatible. And we should be thinking about a plant as a whole and not as one trait at a time. So how do we go about mixing this information, and what is the part that polyploidy is playing? Why is not important in diversification and breeding system is, and how do I combine that information for taxa, right? For taxa, that can be both. So this comes from old analysis that has been very well known for Solanaceae, and big warning here, this might be different for other families, like Fabaceae doesn't have the same system for breeding, but in Solanaceae is very well researched that the breeding system is based on S alleles that are a combination of RNases and pollen that are F boxes. So the way I like to think about it, because I'm a mathematician, is a series of poison and antidotes. So imagine you have antidote S1, right? Sorry, you have pollen S1 that has antidotes for everything else except S1. So when an S1 pollen tries to fall in a stigma that is associated with pollen that is S1, it gets rejected. This is a way of a prezygotic barrier. But when you have polyploid pollen, for example, S1 and S2, and it's trying to fall in the stigma, because it's S1 and S2, it has antidotes for absolutely everything. So polyploid pollen can always be self-compatible. So what it comes from this study from Trala and many others after is that polyploids are always self-compatible, but self-compatible plants can be both diploids and polyploids. So it's not a bidirectional relationship. And then self-compatibles are always diploid, but diploids are can be both compatible or incompatible. So this sets our mind frame to work with new states. For example, incompatible diploid that could have evolved in one step to compatible polyploid via polyploidy, or incompatible diploids that could have evolved to compatibility first and later to polyploidy. So these are the three states that you're seeing here: incompatible diploids in green, compatible diploids in blue, and compatible polyploids in pink. So using these three states, I can define a diversification model that allows me to see what is happening in the mixture of these two traits and noise, right? So that model is what we call the multi-state SSE. So you have your three states: incompatible diploids, compatible diploids, and compatible polyploids. How evolution happens between these states, but each one has its own speciation and extinction. So when we feed this model to the same data set, to the same tree, what comes out of it, and you can see very clearly here, the green posterior distribution of net diversification is much faster than anything that is compatible, either polyploid or diploid. So this plot summarizes what we were seeing partially in the BISSE of ploidy and in the BISSE of breeding system, but by parsing out the effect of combining ploidy with breeding system. So the complex interactions really reveal the difference in diversification. And of course, I did this adding the hidden state, and it comes out very, very similar and a little more complex. If you're interested, I can discuss a little more about it later, but I want to finalize with something important, because you might take from this that maybe polyploidy doesn't matter, and in diversification terms, and whereas that's true, there's an important piece of information that we need to take here. From the rates of evolution between the states, one thing we can calculate using these diversification methods is the pathways to polyploidy. And in these pathways, when diversification is unaccounted for, that is the plot that you have on your left. And this is from a study that Kelly Robertson with Emma Goldberg and others did back in 2011. They found out that it's much easier to evolve polyploidy in a two-step model. For example, going from incompatible diploid to compatibility in diploid, and then to polyploidy. So in the x-axis, you have branch length, and in the y-axis is the a proxy to the probability to have that. And you can see as you increase the branch length, you most likely will see that the evolution to polyploidy is a two-step process. However, when you incorporate diversification in your analysis and recalculate those rates, what we can observe is that no matter at which branch length, we get a more probable evolution directly from incompatible to compatible, incompatible diploid to compatible polyploid, which means that polyploidy is a strong way to evolve compatibility in this system. So I just wanted to conclude with this, because I think that's very valuable, that adding diversification to this whole process can reveal how polyploidy contributes to the evolution of breeding systems. So as I said, just to recap, breeding system is a trait that contains more information about the diversification process in Solanaceae. However, knowing complex interaction of polyploidy and breeding systems can actually tell us how polyploidy evolves and how polyploidy contributes to other traits that are important for diversification. And just from the mathematical or statistical perspective, we need to start considering models that are not single trait, but more, right? And adding complexity to the evolution of polyploids. And the way I like to see it is like these 3D glasses, right? On one hand, you have polyploidy models, and then on the other hand, you have breeding system models. But really what we want is to see the 3D story of breeding system and polyploidy models. And with that, I want to thank my collaborators in this and my funding sources. And thank you, Mike, again, for organizing this.
All right, well, thank you, Rosana, for a wonderful talk. Give everybody a chance to put their questions in the chat box, and then we'll try to have folks ask those questions directly. All right, we'll let Rob Laporte pop up there. Go ahead, Rob, and turn your video on and ask away.
Yeah, so the question is, would you expect similar results if you had a trait other than ploidy? So yes.
Answer it away then. Thanks. Sorry, I didn't know we were doing the video.
Yeah.
We did this last week. It seemed to work a little bit better so we could actually interact. How's that?
All right. Yeah, sounds good. Yeah, I was just curious, Rosana, if I guess I was thinking, does it matter that it's ploidy, or does it have to be similarly structured for how the trait would be acquired? I mean, ploidy is kind of this discrete character, and flower color might be more continuous, I guess. And so would you expect something different, or is that something that could even be parameterized?
So you can measure, so over all this project, one of the big questions is like, how much is the contribution, right? Like when you're doing a regression, you say, this contributes 30% and this contributes 20%. And I was wondering if we could do something like that. You cannot measure the contribution, the percentage contribution itself, but you can measure the relevance of each trait to the process of diversification with something that we call the lumpable models that are in this paper. So you add one at a time, and there's a mathematical way to rewrite it so you can compare the larger model with the smaller model. And when we did that, it looks like if you start with breeding system and add ploidy, it really doesn't add any value whatsoever. But if you start with ploidy and then add breeding system, it does. So it is very asymmetrical, and it's very important to evaluate how you're doing things. Yeah, and there's this work in progress on how you actually measure the contribution of each trait in the presence of others, because they are interacting. I should stop sharing because maybe you're seeing that only my rear screen.
Yeah, sure. There we go. Next question there from Yogendra. Oh, that's to David. All right. Well, let's see if there's any other questions about the Rosana's talk first, and then we'll come back around to David. So Tia Lynn, oh, not Tia Lynn, Fatme has a question for you.
So there is HESI model for continuous trait. Not yet that I know of. Right now, I think the progress on discrete traits is really big, but for continuous traits, there are a lot of problems. To start with, just the basic quasi, that is the model that is used, it's perhaps not consistent on the estimates. So I think it needs a lot more. And if I know, I'll let you know. If I hear something, I'll let you know.
I have a real quick question that I'm going to stick in there real quick. So, you know, we've all been doing these sorts of analyses for a while now, and we've got some similar work, not including breeding system, coming out as well, where we see this kind of discrete structures in the HESI results with polyploidy and other stuff. But the one thing that worries me about everything now is in the aftermath of Matt Pennell's recent paper in Nature, questioning the power of our ability to even see these. You know, we've been doing a bunch of other stuff where we've always kept kind of running Megion and Sanderson's sort of models and setting Extinction, a bunch of things. So I feel like some of the results that we all have generated are probably robust at that level. But how do you interpret sort of all of this whole sort of decades worth of progress in the aftermath of Matt's paper? Yeah.
I've been talking to Matt too because he's my collaborator.
Yeah, exactly. Right. Yeah.
Yeah, so it is very interesting to think about. Well, we cannot estimate any speciation and extinction, right? They are not consistent parameters. In extant phylogenies, you don't know anything about extinction. So, and these are completely correlated. If you run your analysis, you see a 1-to-1 speciation and extinction.
Yeah. Right.
However, this is very, very preliminary. So I've been running some simulations where you assume different things about extinction, like if you know it from fossil records, you have some priors. And what happens is like, yeah, it radically changes the speciation and extinction rate, but the relationship among the two states does not change.
Yeah. The qualitative result may not change.
Yeah.
Right. Yeah.
What it looks like right now is if it doesn't change, if you're willing to assume that extinction is less than speciation, or vice versa, speciation is smaller than extinction.
Right.
It is possible. What my intuition is, and of course this is not complete work right now, but my intuition is that, yeah, perhaps we cannot say, speciation or net diversification is 0.007, but we can say diploids diversify faster than polyploids. And I think that will be really powerful to figure out. But there's other caveats about how they relate, whether a state maybe is different than the other in terms of extinction itself and your prior distributions, et cetera.
That makes me feel better, because that was sort of where our discussion of that paper went as well. And when we play with some of these values in different kinds of models, I don't necessarily, but personally, ever really trust the actual numbers we generate anyway. But it's the qualitative result that you hope is important, right?
The difference on the states is what we want to get at. And another important point here is that Matt, when Matt put this paper, he talks about these congruence classes, right? Like groups of models that are representing similar things. And even though you cannot say this is faster or slower, you can say this is a group. And we yet don't know about what those congruence classes are. And of course, we don't know what those congruence classes are for state-dependent models and diversification. So we still have to figure that out, and in the meantime, we can, one step at a time.
That's right. I really enjoyed that. Looks like there's another question there too, from Zhang Li, and I'll let him pop up and ask that.
How do you think the Asian whole genome duplications in Solanaceae affect the pattern that you see in Solanaceae right now? I mean, I do think that they aid the evolution of compatibility that we see over and over. And I truly believe this is the way to evolve compatibility in Solanaceae. So it might have happened that I haven't looked at the position itself on the branches of the whole genome duplications, but I would assume if you were to position it, you will see a whole genome duplication and then followed by a compatibility switch, which would, and then the diversification, which will be very telling if you were to plot them across the Solanaceae. But I don't have the genomic or genetic data to plot them. Mm-hmm.
That would be fun.
That's a good project for you, Shay.
All right. Well, I think we'll let maybe David had a question there, and maybe we'll bounce back. And there's a couple of questions coming in for David, so let's switch around there and get those on. So I think Fatime had a question here for David. I'm curious if we can do. Let's see. Oh, where are we at here? No, that was a different question. There was another one up there somewhere for David. Oh, here we go. I have from Yogendra. I have a question to David. Does updog can updog be used to identify haplotypes? If not, do you suggest any software?
No, we assume biallelic SNPs, so we're just estimating dosage. I don't know actually what the state of the art is for estimating haplotypes in polyploids right now, actually. So I guess I don't have any recommendations. I guess that's a disappointing answer. Yeah.
You have to have like really good sequencing data, I think, is is probably the good starting point for estimating haplotypes. But yeah. Let's see. Another question for David from George Tiley, if George wants to pop in there and ask that. He's got a long question there. There you are. There's George.
Hi, David.
Hey.
Yeah, maybe I wrote too much, but in general, it's just kind of understanding the updog paradigm, which I like in a lot of ways, and the same thing with sort of like genotype likelihoods through angst. But at the end of the day, there is sort of a lot of software and analyses that don't really handle uncertainty in an elegant way, and they want like hard filtered SNP sets.
Mm-hmm.
And I guess, has there been any comparisons to something like GATK, which can also do polyploid genotyping, but it, it's trained on human data, and that's always sort of concerned me a little bit. So yeah, at the end of the day, is there any real comparison between if we have to start taking hard cutoffs and how that looks between different polyploid genotyping approaches?
Yeah, that's a great question. So I don't think there's been a lot of exploration on the downstream effects of the different genotyping softwares. Like most of the comparisons happen just to see which ones do the best at actually estimating the genotypes, rather than look at the big effects on downstream things like mapping or GWAS or whatever.
Mm-hmm.
My intuition is that if the average behavior is what's important across many SNPs, then it's less important than if it matters like at a particular SNP. Like if a particular SNP's behavior is very important, then there's probably a lot of difference between these approaches. But if it's the average behavior across many SNPs, that's important, then there's less of a difference. But I don't think people have done a comprehensive study of that.
Okay, interesting.
All right. Any last questions there from anybody before we call it a day? All right. Well, I want to just thank both David and Rosana for two wonderful talks this morning. And I want to highlight that our speakers in 2 weeks we'll hear two more great talks. First up will be Sonia Garcia telling us about B chromosomes, sex chromosomes, and polyploidy, which should be really exciting, I think, to hear about some of the different sort of chromosomal diversity that we have in genomes and how that interacts with sexual systems and ploidy level. And we'll also hear from Joel Charbrough, who will tell us about cytonuclear evolution and allopolyploids. So tune in again in 2 weeks for that. And with that, I'm going to stop recording and just thank everyone for attending today, and invite everyone to stick around and feel free to chat and ask any other questions and just talk about polyploidy if they want, or whatever else is on their mind for a little while. But thank you to our wonderful speakers and everyone for participating today.